Hi guys, this is Ms. Romani, and this week we're going to be learning about asexual reproduction and a type of cell division called mitosis. Let's start by talking about reproduction. Now, life is extremely dependent on reproduction and a related process called cell division, because reproduction is basically the only way organisms have for passing on their genetic information from one generation to the next. Also, if you think about it, without reproduction, life would cease to exist. There are two types of reproduction: asexual reproduction and sexual reproduction. Before we begin our lesson on asexual reproduction, I'd like to tell you a story. This is the story of the modern banana. The modern banana is both a miracle of biology and an incredible biological disaster. First, unlike most other fruit, there's mostly one type of banana that gets eaten all over the world. It is the Cavendish banana. If you have bananas at home, there is a 99% chance that it is a Cavendish banana. And this banana is a clone. Every one of the 100 billion Cavendish bananas eaten throughout the world every year is genetically identical to each other. So why are bananas clones? Well, let's think for a minute about how we get plants. We need to plant seeds. Seeds grow in the part of the plant that becomes the fruit. But bananas have no seeds, so you cannot grow new banana seeds using the bananas that we do eat. Wild bananas, on the other hand, do have seeds. But of course, as you can see in this picture of a wild banana with seeds, this would be very difficult to eat. So through the process of domestication, people found a way of developing seedless bananas. To do this, we use a process called vegetative propagation. No seeds are planted. All you need is one banana plant, and at the bottom of the banana plant, tiny, smaller stems grow out called suckers. And so if a sucker is removed from the base of the banana plant and planted somewhere else, the sucker will actually grow into a new banana plant and will eventually produce the fruit, the banana, but it would be seedless. This new plant is genetically identical to the parent plant, and that's how plantations are made. Suckers are removed from plants and planted somewhere else, and eventually you end up with what is called a monoculture. And that's when all plants in a farm or a plantation are genetically identical to each other. So back to the Cavendish banana. It turns out that when your grandparents were young, or at least most of your grandparents, depending on how old they are, the Cavendish banana was not the banana. Up until the 1950s, the one banana that was sold worldwide was a much better tasting banana called the Gros Michel or Big Mike banana. So what happened? Sadly, it was killed off by a fungal infection called Panama disease. It turns out that growing a monoculture comes with a very significant biological danger, and this is because of a lack of what we call genetic diversity. Since every banana plant is essentially genetically identical to each other, a disease that kills one plant will also kill the rest. If one plant is killed by a disease, it means that it has no protection against the disease, so all its clones will be similarly affected by the disease. So Panama disease started to actually wipe out banana plantations as early as 1890. Seen from above, the plantations across Latin America, Africa, and throughout the world started to look like just lights had been turned off. Patches of bright green went black, whole landscapes went black within a year of a disease arriving at a plantation. And once devastated, plantations had to be abandoned because it turns out that the fungus that causes the disease can survive in the soil for decades. So by the 1950s, all plantations worldwide were pretty much destroyed. So banana corporations were finding themselves in big trouble. They had to try to find a solution or risk going out of business. And the fungus could not be destroyed, no matter what they tried. So the only thing to do was just to find another banana, one that was actually resistant to the fungus so it could be planted on the abandoned land. And the only banana that seemed to be both resistant to the fungus and similar to the Gros Michel banana was a banana that came from a greenhouse owned by Sir William Cavendish, a British duke. And this was the Cavendish banana. Unfortunately, according to accounts, I've never tasted a Gros Michel banana, but according to accounts, the Cavendish banana tasted very different than the original Gros Michel banana. It was much less sweet, and consumers were initially not happy with the change. But by 1965, there was no choice. The Gros Michel banana had disappeared completely. The Cavendish banana could be planted in the same plantations where Anaphase disease fungus was still present, so it became today's banana. And that would be the end of the story, except for one thing. This is not a happy story. Remember the slides? These are not from the 1950s. Those photographs were taken last year in 2019. As a matter of fact, since the 1990s, a new strain of the Panama disease fungus has been successfully destroying Cavendish banana plantations worldwide. The disease has been named TR4 for Tropical Race 4. Scientists have been hired by banana corporations to try to combat the disease, but they're losing the fight. The disease is extremely difficult to fight, and once it arrives in one plantation, essentially will destroy it within a year. So one solution that scientists are looking at might involve actually genetically modifying the Cavendish banana in order to make it resistant to the fungus that causes the disease. But another solution is to find a new banana. So just like your great-grandparents and grandparents grew up with a banana that you never got to taste, your children may also never get a chance to taste a Cavendish banana. There are a few banana breeds that are candidates for becoming a new banana. There's the Lacatan banana, a very popular banana in the Philippines. It has a good amount of beta-carotene, which gives it a sort of a yellow-orangey color. There's the apple banana. Apparently, this banana has a creamy consistency. And very interestingly, when young, they taste tangy with notes of apple. But when ripe, they become more tropical with hints of pineapple and strawberry. And there's also the Goldfinger banana. And some say that this is actually the best candidate to replace the Cavendish because of its pest and disease resistance. It is especially resistant to the fungus that causes Panama disease and TR4. So this might become our new banana. I've never tasted a Goldfinger banana, but apparently it tastes pretty good, but it has a slightly acidic taste. So this might be a future banana. I mean, the Cavendish banana has not disappeared yet, but the fight against Panama disease, it's not looking good. The story of the banana can serve as a warning to the drawback of what we call asexual reproduction, which is really what our lesson is about. So let's get back to our lesson. Asexual reproduction involves basically a single parent. Offspring are genetically identical to that one single parent, and they're essentially clones of that parent. There are a few ways that asexual reproduction can occur. The simplest just involves cell division. One cell divides into two identical cells. Of course, this is something that is done by single-celled organisms. So this type of asexual reproduction is common in protists and bacteria, and it involves just one single cell dividing into two after making a copy of their genetic material and making sure that each new cell gets a copy of that genetic material or DNA. The process is called binary fission, and here you can see in this first picture a paramecium, which is a eukaryotic protist that is just separating into two cells. This is a slide showing bacteria reproducing through binary fission, a form of asexual reproduction. Another form of asexual reproduction we've seen already is something called vegetative propagation. This is common in plants. Vegetative propagation, like all asexual reproduction, involves only one parent plant, and the new plant is genetically identical to the parent. It can happen naturally with some plants, but we most often see it when humans help it along. There are many ways that vegetative propagation can happen. For example, with bananas, a sucker is removed from the parent plant and transplanted. With some other plants, you can have something called a runner, which is what happens when a stem that's still attached to the parent plant grows low to the ground, and then it grows its own roots. And then the runner often remains attached to the parent, but if it's separated, it has its own root system, so it's basically its own new plant, but it's genetically identical to the parent plant. Other methods sometimes might involve taking what farmers call cuttings of a plant. These are pieces of stem or roots that can then be transplanted somewhere else. Gardeners often do this in order to move plants or propagate plants that they really like into new areas of a garden or into new gardens altogether. Some animals also reproduce asexually. For example, starfish reproduce through a process called fragmentation and regeneration. So what happens with starfish is that this is a process of asexual reproduction that involves cutting body parts. And then the organism will regrow the body parts that were cut, and the body parts that were cut will regrow the rest of the organism. So for starfish, for example, if you cut out an arm of a starfish, that arm will regrow the rest of its arms into a brand new starfish, and the original starfish that lost the arm will regrow that arm itself. So technically, a starfish has 5 arms. You could actually grow 5 starfish by just cutting off each individual arm, and each arm will regrow a new starfish around it. This is not the main way by which starfish reproduce, by the way. They actually reproduce sexually, but they can reproduce asexually through fragmentation and regeneration. And of course, under this process, you don't need 2 parents. You just need the 1 parent, and every single starfish that results from this type of reproduction is genetically identical to the parent starfish. Another animal called a hydra, which is a tiny aquatic animal, reproduces by a process called budding. Budding basically involves the growing of a baby or an offspring that is genetically identical to the parent off of the body of the parent. So the hydra, for example, grows a young baby hydra off of its body, and then eventually that bud breaks off and can grow into an adult hydra that is genetically identical to its parent. And more interestingly, some lizards and other reptiles and some amphibians have a way of reproducing asexually through a process called parthenogenesis. So parthenogenesis basically involves species of certain animals, like certain reptiles or amphibians, that have only females in the species. And the females will lay eggs, and the eggs will be clones of the mother. And so they would involve absolutely no sexual reproduction, no males, and no fertilization. Just females that lay eggs to their own genetic clones. It's rare, but there's a few examples of it. One of the most famous examples is a whiptail lizard, which is a lizard that is common in the deserts of Arizona and New Mexico that have basically just all females in the species. There are no males, and they reproduce asexually through parthenogenesis. Humans and most plants and most animals do not reproduce by asexual reproduction, but we do have to produce genetically identical cells. And we do this all the time. We're constantly replacing our cells. We're constantly undergoing cell division. Some say that we replace most of our body cells every 10 years. So let's talk about that for a minute. Let's talk about the process of cell division. So when we talk about cell division, we're referring to something called the cell cycle. The cell division part of the cell cycle is only a small part of that process. The cell cycle is basically a continuous sequence during which cells in our body grow during a stage called interphase and then divide through a process called mitosis. And the function of cell division and mitosis can be threefold. The first one, of course, like we discussed, is reproduction. For certain asexual organisms, like say the hydra or the starfish, the way that the hydra makes its little bud is through mitosis, cell division. It just makes extra cells, and the cells eventually become its bud and then breaks off. Or the starfish will regenerate its arm, and the arm will regenerate the rest of the starfish through cell division called mitosis. For most plants and animals that reproduce sexually, cell division is basically there for growth. This is how we grow. We may need to make new cells so that we can grow bigger. A baby does not become adult size by having their cells become bigger. A baby becomes adult size by growing more cells, by cell division. We all actually started out as a single cell called a zygote. And then that single cell divided and divided and divided until we became an actual baby that was born. And by now, there are trillions of cells in your body, and you got those cells through cell division. The other reason why we need to undergo cell division is because we need to repair our body tissues. We're constantly replacing old tissues, worn out tissues, and repairing damage that is done through injury and as we age. So we need to make new cells to repair damaged tissue or replace those worn out cells. If you break a bone, the bone gets set and eventually gets healed by cell division. Bone cells reproduce, make new bone cells, and basically seal the cut. You break your skin open and you get a cut, same thing happens to your skin. So let's talk for a little bit about the timing of some cell division that happens within our body. Because even though we're constantly replacing our body cells, this replication of those cells, the replacement of those cells, does not happen at an equal timeline. So for example, to replace the cells that are in our stomach lining, in our intestine, in our gastrointestinal tract, would take about 1 to 3 days to replace. And that's because this area of our body is under constant stress. I mean, we have food moving right through it, so the lining of our intestine and our stomach is constantly being replaced, and our skin gets replaced every 20 days. Not all at once. We're not snakes. We don't shed our skin all at once. It's not like we walk around on day 19 looking horrible and then emerge fresh underneath our skin on day 20. That'd be cool. That's not how it works. At this time right now, all over your skin, you have some cells that are on day 1, some that are on day 5, some that are on day 20. And we basically are constantly just replacing our skin cells. When our skin cells die, they fluff off into the air, or if we shower, they go down the drain in the shower, or we take a bath, they end up in the water in the bath. A lot of the dust that we see in our homes is essentially the old skin cells from your body and your family's bodies that are just in the air. Our blood is replaced every 120 days. So if you've ever donated blood or if you've ever received blood, a blood transfusion, you're thinking, well, will you always have someone else's blood in your body? No, that's not the case. Our blood cells are not there forever. They get replaced every 3 months or so. So we are constantly replacing our blood cells. Our immune cells will take about, depending on the type of immune cell, between 10 hours to years for them to undergo cell division. So we have some immune cells that are in our body for years. Those are the cells that carry our immunity, and some of them are just like some white blood cells might only last a few hours. Fat cells we replace every 10 years or so. Bone cells might last between a few days to 25 years, depending on the type of bone cell that we have. And finally, brain cells and nerve cells. This is the one that is the longest lasting out of all the cells, and they pretty much last, some of them, a human lifespan. We are born with almost every brain cell that we'll ever have. We actually still continue to grow our brain for the first 2 years of life. So this is why they say that it's extremely important to make sure that we feed babies a good diet, because for the first 2 years of life, we're still developing our brain and actually growing our brain. And after that, our brain can still make connections and we can get smarter, but we don't grow new brain cells. And sometime around the age of 18, 20, 25, then the process will start where brain cells will slowly start to die. And so when brain cells die, unfortunately, that's it. We don't regenerate them. They don't divide again. This is why brain injuries can be so devastating, or spinal cord injuries as well, because nerves are also, especially our spinal cord nerves, they do not divide. So any injury does not get healed. Unlike breaking a bone, where bone cells can sort of divide in order to heal, the same is not true of nerve injury. This is the first few days of a tadpole's life. And as you can see, at first, the cells just get smaller and smaller and smaller. This is the very beginning, and eventually it's going to go through a process where the cells will actually get bigger as it divides. At first, the initial cell is really big, and then cell division is happening, and you end up with more and more and more cells, and then, soon enough, it's going to slow down a little bit. The cells will get bigger, and before they start to divide, and then they'll start a process called differentiation, where the cells will, some will become skin cells, some muscle, some eye cells, brain cells, and so on. So we'll talk about the cell cycle, and we'll talk about the stages of it now. So there's the first stage, and this is the stage where the cell is just growing, living its life, doing what it needs to do. It's called interphase. And cells, basically, depending on the type of cell, will spend most of their life on interphase. Brain cells, for example, will spend 85 years on interphase. This is the stage where the cell is just busy living and doing its job. Intestinal lining cells might spend maybe 2 days on interphase and then 1 day on dividing. This is the stage that is usually the longest time span. And interphase is divided up into 3 stages or 3 phases. At the beginning, it's called the G1 stage. The cell just grows, does its job. Then we end up with the S phase. This is where the cell continues to grow, but now it's also busy making a copy of its DNA. And that is because when the cell divides, which is the next stage after interphase, we need to have 2 copies of the DNA so that each new cell is genetically identical to the original cell. Every cell needs to have all the DNA. So before a cell divides, it needs to make a copy of the DNA. And so that happens during interphase, before cell division even begins. And then the last stage is called the G2 stage. And this is when the cell is now going to grow in preparation for cell division. It's going to grow bigger and make any final preparations that it needs in order to actually divide. And so the division stage is also called the mitotic stage. And it's the shortest event of the cell cycle. And it involves mitosis, which is the division of the nucleus. And it's separated into 4 stages as well, which we're going to get into. And cytokinesis, which is the division of the cytoplasm and the rest of the cell. So all the rest of the organelles that does not include the nucleus. So to recap, most of the time the cell is just growing and doing its job, doing its thing, being a brain cell, being a skin cell, doing skin cell things. Then it starts to get ready for cell division. And so still during interphase, it will make a copy of its DNA, it will get bigger, and it will prepare for cell division. Then it enters what is called the mitotic stage, the division stage. And for that stage, there's mitosis and cytokinesis. So I'm going to give you a little bit more detail about what happens during each of these stages. So I have this animation here that will go through the different stages of mitosis, starting with interphase, moving all the way through all the different stages of mitosis, which we call PMAT for prophase, metaphase, anaphase, telophase. And so I want you to focus on what's happening to the nucleus and more specifically what is happening to the chromosomes. So we start with interphase. There's the cell. You can see the nucleus is right there in green. And the first thing that's going to happen is the nuclear membrane is going to disappear. And when that starts happening, that's prophase. And then metaphase, anaphase, telophase, and cytokinesis. And I know it was too fast. So let's start by looking at interphase. So interphase, like I said before, this is the stage where the cell is just growing. This is not the cell division stage. This is the stage that the cell spends most of its time just doing its job. But prior to cell division, during the G2 phase of interphase, the centrioles in animal cells will double. Plant cells do not have centrioles, so this does not happen in plant cells, but in animal cells, the centrioles, which play a role in cell division, which you'll see in the next few slides, they make a copy of themselves because you need 2 sets of centrioles. The other thing that you will notice during interphase is that the nuclear membrane is present, the nucleolus is present, the chromosomes are not present or visible yet. DNA is organized as chromatin at this stage, so like that loose sort of stringy spaghetti. But by the G2 stage, the chromatin has made a copy of itself. The DNA has replicated. That happened during the S phase. So during interphase, chromatin does make a copy of itself. The DNA replicates, and it starts just preparing for cell division by making copies of centrioles and sometimes also making copies of other organelles. So this is what interphase looks like in an actual cell under the microscope. So it's a little bit more difficult to obviously recognize the different stages when you look at them under a microscope for a real cell as opposed to just a picture that is drawn. But for interphase, in order to recognize it, what you're looking for is you're looking for a fully visible nucleus. So you can see the nuclear envelope, you can see the nucleolus. You do not see individual chromosomes. The chromatin just looks like a shadow. Now, prophase is when we begin mitosis, and this is the beginning of cell division. Prophase starts by the centrioles moving to opposite sides of the cells, and these little fibers, protein fibers called spindle fibers, start to appear in between and shooting out of the centrioles. The chromatin will start to condense, and the chromosomes will now become visible, and the nuclear membrane and the nucleolus both disappear. And so now you start to see the crumbling of the nucleus. The nucleus will reappear by the end, but during cell division, the nucleus is gone. And the chromosomes remain, and they will become more visible as they become condensed. This is what prophase looks like under the microscope. It's a little bit different. You can have different appearances of prophase depending on whether it's early on, like in the picture on the left there, you see that one pointing to early prophase. You can start to see that the nuclear membrane is starting to fall apart, and then by late prophase, the nuclear membrane is completely gone. The key with prophase is that you see the chromosomes as more condensed structures as opposed to the sort of blurry shadow that you see during interphase, and that you either see the nuclear membrane disappearing or no nucleus at all. The next stage is the metaphase stage. And metaphase is distinctive because by this point now the spindle fibers have attached themselves to the chromosomes, to the centromeres of the chromosomes. Spindle fibers are made out of proteins called microtubules that essentially act like rope. And they hook on to the centromere of the chromosomes and they move them around. And what they do, they've actually moved the chromosomes so that they are not just gathered in the middle of the cell where the nucleus was, but they're all lined up along what is called the equator of the cell in a single file going from one end of the cell to the other, and just line them up in the middle, all one by one. So the centromere is the place where the spindle fibers attach. The spindle fibers are those protein fibers that are shooting out of the centrioles, and you can see the chromosomes very visible. Now, even under the microscope, metaphase is actually one of the easier ones to recognize. You can see right here on both the right and the left pictures. The one on the right, by the way, the one that looks more like a circle is an animal cell. The one on the left is a plant cell. You can see the chromosomes sort of lined up right in the middle of the cell. The next stage after that is anaphase, and what is going to happen during anaphase is that the spindle fibers that are still attached to the centromeres are going to start to shorten. And as they do, they're going to split the centromeres in two. And so what will happen then is that the sister chromatids, the identical copies of the DNA that you would find in the chromosome, that are going to be moved to opposite poles of the cell, and they're going to be moving towards the centrioles. So the chromosomes were lined up at the equator, and now they're moving towards the poles of the cell. As the centrioles split, because the spindle fibers that are sort of holding onto them are tugging on them, and then they're moving to opposite sides. And then this is what anaphase looks like under the microscope. You can clearly see that the chromosomes are now split and moved to opposite ends. Now you can see also some spindle fibers in the middle and between, and that is because there are also some fibers that are growing in between the two poles to just sort of make the cell a little bit longer. And finally, we have telophase. And telophase is going to be the final stage of mitosis. Mitosis is all about dividing up the DNA from one nucleus into two nuclei. And so it's going to be about reforming the nuclei. And you'll find that it's a lot of the same steps that happen during prophase, but opposite. So for example, the spindle fibers now disappear. They're no longer needed. Once the chromatids have moved to opposite poles, the spindle fibers will disappear. The nuclear membrane will reappear around the chromatids that are now on each new end of the cell. The nucleolus will also reappear. The chromosomes will then uncoil and become chromatin again. And while telophase is happening, a process called cytokinesis is also happening. So cytokinesis is essentially the division of the rest of the cell, but it's happening at the same time as telophase and the formation of the two new nuclei in each of the new cells. So during cytokinesis and telophase, you cannot tell one from the other. So when you look at them under the microscope, you find a cell that is under cytokinesis, it is simultaneously undergoing telophase, and vice versa. So let's take a look at what happens during cytokinesis. You can see it's the same picture because this basically happens simultaneously. So during cytokinesis, this is when we describe what is happening to the cytoplasm and to the organelles and to the membrane and cell wall if you're talking about a plant cell. So the cytoplasm and the organelles divide. For animal cells, as the cell gets bigger and elongated, a cleavage furrow will form in the membrane in the middle, which is essentially what we call when the membrane starts to pinch inward and eventually meets from the top to the bottom and splits into two cells. And we call that indentation, we call that a cleavage furrow. Plant cells, on the other hand, do not form a cleavage furrow because they have a cell wall. And so what needs to happen is the formation of a new cell wall. And so a plant cell will get longer and the cell wall will form right in the middle in pieces at first that eventually just become one solid wall. That cell wall that is in construction is called a cell plate. So when the cell wall is built, we call it a cell wall, and it's now two separate cells that are attached because plant cells sort of attach to each other. When it's forming, we call that a cell plate. And that's that. And so as you can see at the top, you can see all the different stages, what it looks like when we draw a picture of it, and then underneath what it looks like when you actually see it under the microscope in real life. Next, we're going to learn about sexual reproduction in meiosis, which is a very similar process to mitosis. So a good understanding of mitosis will certainly help understand meiosis. And so that's it for this lesson. Have a great day, everybody.